All right, tumor suppressor genes. Just a quick side note about tumor suppressor genes. The way to form neoplasias is either by increasing proliferation or decreasing the suppression. So, speeding up the gas or taking your foot off the brake. So we're going to talk about taking our foot off the brake pedal now. Let's discuss briefly, because it's important, let's discuss the number of mutations that need to occur for a neoplasm to form. So, recall that we have 2 chromosomes for every gene, right? 2 loci for every gene, 2 alleles. So, you get each chromosome from a different parent. And here's one allele and here's the other for the same gene. Now, for oncogene, because we have 2 of them, if you have a mutation in one of these genes for an oncogene, that's all it takes. Boom, you start making more MYC, MYC, or any of the other oncogenes, you're going to have the progression to cancer. That's all it takes. All you need is 1 chromosome involved. But for tumor suppressors, if you have a mutation in your repressor gene on 1 chromosome, you lose that gene, you still have the other one, right? You still have that one that's going to be suppressing the transformation of neoplasias. So you need to take out both genes for cancer to develop in the case of tumor suppressor genes. Does that make sense? So you only need 1 mutation for oncogenes and 2 for tumor suppressor genes. Very good. Now that we got that sorted out, let's keep going with tumor suppressors. So we talked about the RB protein. We talked about how once it's phosphorylated, it leads to the freeing of E2F. Okay? E2F gets freed up, and it allows the progression from what phase to what phase? The G1 to the S phase. Well, there's another guardian of the G1 to S phase besides RB, and I just had it up here briefly, but what is that one? It is the P53 protein. Okay, and by the way, what does that do? So, essentially, P53 on chromosome, do you know what chromosome it's on? Chromosome 17, and by the way, RB is on what chromosome? Chromosome 13. Okay, so chromosome 17, it acts as a gatekeeper of the cell, and it ensures that all mutations are corrected within the cell before it can progress to that synthesizing of DNA phase, the S phase. Otherwise, if it's unable to repair all the mutations, P53 will induce, well, what kills the cell? When does the cell kill itself? Apoptosis. Through activation of caspase enzymes, cytochrome C from the mitochondria, you have apoptosis taking place. All right. So let's talk about, real quick, we forgot to mention some of the cancers associated with RB. P53 is essentially with every type of cancer. RB, well, what does it stand for? Retinoblastoma. So obviously it's associated with retinoblastoma. And then we mentioned the other one. Do you remember what that is? Osteogenic sarcoma of the bone. Very good. All right, so let's talk about some more tumor suppressor genes. So when DNA gets broken, there's a certain protein that can repair this broken DNA, and that is BRCA, or BRCA. And I think of this as BRCA repairs broken DNA. So that helps me remember it. So that's a good mnemonic, but what does really BRCA stand for? Breast cancer, because it's associated with breast cancer. And there are 2 main types of BRCA. BRCA1 is located on chromosome 17, whereas BRCA2 is located on chromosome 13. And those are both associated with breast cancer and a different type of cancer in women? Ovarian cancer. Good. All right, let's now talk about the RAS protein again. So we mentioned this earlier. It's associated with colon cancers. And recall that it's activated by GTP, right? GDP transforms into activated GTP, and then you have downstream effects, the MAP kinase cascade, etc. So if this is mutated, if there's any mutations involved in this process, you can run the risk of cancer. Now, there's actually a BRAF protein that regulates the MAP kinase cascade. And if this is mutated, you can develop a certain type of cancer. What type of cancer is that? Melanoma. All right, what other protein prevents melanoma? Just off the top of your head, do you know it? P16. It's good to know. All right, now, speaking of RAS, what prevents RAS from always being activated? There's a GTPase that actually degrades GTP and turns it back into GDP. And this is associated with what genes, chromosomes, diseases, and cancers? That's a lot of stuff to know, but do you know it? So neurofibromatosis type 1, which is on chromosome, so NF1, on chromosome 17, that's associated with neurofibromatosis type 1, hence its name, and increased neurofibromas. There's other signs too, like Lisch nodules in the iris of the eye, plexiform lesions, café au lait spots, means coffee with milk appearing spots, patches all over a person's skin, and their skin can look like elephant hide with these neurofibromas. So you'll recognize it. You'll learn this in great detail later. Now there's another type of neurofibromatosis, type 2 on chromosome. So type 1 is on 17. Type 2 is on chromosome, it's easy, 22. So NF2 on chromosome 22. All 2s. And then another thing to know about 2s is there's 2 tumors in the 8th cranial nerve. So what's the 8th cranial nerve responsible for? Hearing, right? So acoustic neuromas or schwannomas bilaterally occur in NF2. And both of these are associated with a GTPase not working. So the GTP is constitutively activated, and the RAS protein thus is always telling the cell to grow and divide.